Hello friends, in this video we are going to discuss about post-transcriptional processing in eukaryotes. So first of all, what is post-transcriptional processing or its definition? It is a set of biological processes of eukaryotic cells which chemically alter primary transcript, that is RNA. Here we will mainly focus on messenger RNA to produce functionally mature RNA is called post-transcriptional processing. It takes place prior to translation, that is protein synthesis. And for this, messenger RNA is transported to cytoplasm. So, post-transcriptional processing occurs before being transported from nucleus to cytoplasm for translation or protein synthesis. It includes three processes: capping, tailing and splicing. Let us see them one by one. First is capping. This is the first processing event. It occurs at 5' end of RNA where 7-methylguanosine triphosphate is added, and this process is catalyzed by enzyme guanyl transferase. So you may think that why capping is required or what is the importance or functions of cap. Cap guides the messenger RNA from nucleus to cytoplasm where translation occurs. It gives stability to the primary transcript. That is, it protects messenger RNA from degradation by RNases present in the cell. In this way, it increases the half-life of messenger RNA. It also helps in recognition of messenger RNA by translation machinery, which is important for the translation or protein synthesis process. It helps in the formation of messenger RNA ribosome complex. It helps in the binding with smaller subunit of ribosome during translation initiation. This is because this cap is identified by 18S ribosomal RNA of smaller subunit of ribosome. It is also binding site of many proteins involved in translation. Now, second process is tailing. It is the addition of adenine residues, about 200 to 300 at 3' end in a template independent manner on newly formed HN RNA. In this, template is not required. It occurs independently. And this process occurs with the help of poly-A polymerase, and this is called poly-A tail. So this process is catalyzed by poly-A polymerases, and 200 to 300 sequence of this adenine residue that is added at the 3' end is known as poly-A tail. And this process of tailing is also known as polyadenylation. Now, functions or importance of tailing. Polyadenylated tail gives extended stability as it protects messenger RNA from degradation by RNases. And these RNases are present in the cytoplasm of cell. This is because messenger RNA is transported from nucleus to cytoplasm as translation, that is protein synthesis, occurs in the cytoplasm. It signals the end of transcription to the cellular factors. So to the cellular factors, this poly-A tail gives the signal that transcription is ended or transcription is over. It helps in the movement of messenger RNA into the cytoplasm. Third is splicing. It is the process of removal of introns which are non-coding sequence and reattachment of the exons which are coding sequence into a continuous molecule. Reattachment occurs in a defined order. It takes place, or the splicing takes place by spliceosome machinery, and this spliceosome machinery consists of small nuclear RNAs, snRNA and proteins, and these spliceosome machinery only recognize the splicing sites. Now, the need or functions of splicing. It helps in evolution by creating new combinations of exons by alternative splicing. So, by alternative splicing, splicing occurs in different manner. Hence, different combinations of exons are created. and this helps in evolution of organism. It also helps in regulation of gene expression and protein content of the cell. Now let us understand post-transcriptional processing with the help of this diagram. This is pre-messenger RNA which has exons and introns. Exons are coding sequences and introns are non-coding sequences. So in the first step, cap is added to the 5' end. This cap is 7-methyl guanosine triphosphate. Then in the next step, polyadenylation takes place at the 3' end. That is 200 to 300 adenine residues are added to the 3' end. After that, splicing takes place. It means introns, which are non-coding sequences, are removed and exons, which are coding sequences, are reattached. So at the end, we get mature messenger RNA, which has cap at the 5' end and poly A tail at the 3' end, and it has coding sequences known as exons. This entire process takes place in the nucleus, and then the mature messenger RNA enters the cytoplasm. So what we can conclude, that fully processed heterogenetic RNA, which is called messenger RNA, is then transported out of the nucleus to the cytoplasm for translation, which is known as protein synthesis. This translation we will discuss separately in detail in series of videos. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.